Welcome, everyone. Great to see a good live audience again, and hopefully also a good online audience. After the excellent presentation by Shangsong Zhu yesterday, we have another highlight today. So it's one after another. And we're really, really pleased and honored by the visit of Markus Kaiser. And I didn't realize that he hadn't been to Hamburg yet. So this is a grand omission, and I'm very glad we're correcting it now. As some of you, probably many of you know, there's a special relationship between Markus and myself. And as the word goes in German, the Doktorvater is the supervisor of the PhD of the graduate student. And so it fills me with fatherly pride to look at the amazing career of Markus Kaiser.
Your brothers.
Or maybe stepbrothers. But I should say that this is sort of maybe a misplaced pride, because I mean, Markus did it all by himself. And just to mention as an example, I mean, he studied biology and computer science, and his very first paper as a master's student from Münster University with Markus Lappe was already getting published in Neuron. So I mean, yeah, I can be proud of these things, but it wasn't me, it was all him doing these wonderful things. But we had a really great time. In fact, Markus was the first graduate student I ever had, and it was a really exciting time then at a fresh startup university in Bremen. And we were still remembering these long nights that we spent there, just sustained by pizza from the local delivery service and shooting ideas. So that was really fun. Markus then went to Newcastle on a graduate fellowship and very quickly rose through the ranks of the faculty there, eventually becoming professor of computer science and holding affiliation at several schools, I think, before then moving to Nottingham in 2021. Where he joined the imaging center, and that's affiliated with the medical school. So that's, I think, part of his desire to also implement his many ideas in a more practical setting and to give them clinical applications. Markus has published too many publications to count here, many of them in top journals and on very diverse fields. So starting with all the ramifications, interpretations of connectomes, pioneering many of the approaches that people use in the field now on lesioning and perturbing connectomes in order to understand their structure better. Then moving on to the field of developmental connectomics, which was really very fresh in the zeros. And again, some of the pioneering papers that get cited all the time when people want to talk about generative algorithms of network development are due to Markus. Before then moving on to Changing Connectomes, which is also the title of his book, published by MIT Press in 2020. And I can highly recommend it. It's a wonderful read and very, very stimulating. And now he's actually moved on to even more practical things. So if you want to know about the challenges of a theoretician becoming an experimentalist, there's a 10 rules paper in First MD that tells all about that. And I think it's in that context that Markus will talk today about changing connectomes towards neuroinvasive brain stimulation techniques to improve cognitive performance. And we're very much looking forward to that, Markus.
Okay, thanks very much for the introduction and for the invitation to give a talk here. It wasn't all by myself doing the PhD, so I had big help from Klaus. So I will talk about how we can use information about brain connectivity to inform interventions in patients, in particular mental health. So we can basically look at how the brain is connected. You see structural connectivity here, connections over fiber tracks between different parts of the brain. And we can use this image to already predict age plus minus 8 years if you go from a range from 4 years to 80 years, or IQ plus minus 3 IQ points. So just looking at the structure of the network can already give us some information about individual participants. But we want to use this information in order to develop novel interventions for mental health. As Klaus mentioned, there's more information about development in the book here, healthy development for different brain disorders during early stages and during old age, and different mechanisms how to generate networks, highly connected nodes and modules and cortical layers and so on. But today I will focus on connectome applications related to brain stimulation as an intervention for mental health. The long-term goal is to change brain connectivity. If you think about brain stimulation, you think about immediate effects. So you might get a reduction in tremor for Parkinson's disease, or you might want to stop epileptic seizures for epilepsy patients. But I'm thinking about long-term applications of brain stimulation. The idea is to stimulate once or in a couple of sessions and then have a long-term change in brain connectivity so people don't have seizures anymore, or people don't have psychotic episodes anymore. So it's a long-term change of brain connectivity that I'm aiming for. So we're not looking at brain stimulation applied to the time frame of seconds or minutes. So we look at long-term changes of the system over days, weeks, and years. And what are the challenges of brain stimulation in order to lead to long-term changes? One challenge is to decide where to stimulate. Okay, so depending on how we parcellate the brain, we might have around 100 regions or so, or 1,000 fiber tracts. So which one should you target for brain stimulation? That's one of the challenges where computational modeling could get into. Then how to stimulate, determining the personalized optimal stimulation protocol. So for each individual, there might be a need for different protocols, duration of stimulation, the frequency of how many sessions you need to have, and so on. We would also ideally like to estimate long-term effects and unwanted negative side effects. What happens for implants is that they might work initially, but then after a couple of weeks, they stop working and you have to readjust the parameters. So it would be useful to have computational models that can predict what the optimal parameters are, so you don't have this kind of trial and error period. So you see one example here for major depression, where you have this kind of parameter optimization phase, this trial and error phase to find out what actually works for each participant. So it would be useful to have computational models before that. And one main objective is to have an approach that is non-invasive, because people really don't like implants. This is very different from what you hear in the media. You hear about Elon Musk's Neuralink and all those exciting novel technologies that they want to put into your brain. But people don't like that. So even if you think about minimally invasive implants like cochlear implants, only 5.6% of patients who would benefit from a cochlear implant actually agree to have one. Or even if you think about something very, very non-invasive, to have those RFID chips in your hand, like size of the rice corn, very few people are happy to do that. So we did a survey of healthy participants and patients to ask them about their opinions about different interventions for brain and mental health conditions. So we were asking healthy participants, if you would have a brain or mental health condition, what kind of intervention would you like to have? And we were asking patients as well. So in total, almost 800 participants, around half or so were patients. You can see the distribution, a lot of patients with movement disorders, but then we have mood disorders, depression as well, and some other disorders. And they could then rank different options for intervention. So on the right-hand side, you see the preference, which technique they would prefer for an intervention, and ultrasound was number one. And for many people, ultrasound was the first choice as a technology. People possibly think about pregnancy scanners. So if it's good for a pregnancy scan, then it should be okay for the brain. Then you have magnetic non-invasive stimulation, pharmaceutical drugs, electric non-invasive stimulation. People possibly thought about electroshocks and watching One Flew Over the Cuckoo's Nest. So not in favor of that. And people really don't like implants. Okay. So implants are not first choice, but they're basically, for most of the people, they are kind of final choice as a treatment. If we then look at safety, ultrasound is perceived as safe as well. Also perceived as effective, but not as effective as potentially other technologies. So the reason why people are in favor of ultrasound is more about the safety rather than the efficacy of the technology. So implants might be more effective, but not as safe in the perception, and that's why ultrasound is number one. So we established this network in the UK, where we basically bring clinicians and experimental researchers and computational modelers and charities together to look at novel technologies for non-invasive brain stimulation. So there are different people evolved from Imperial, Oxford, Nottingham, and Edinburgh. We had several funding calls where we have pilot projects to establish novel technologies. And the idea is really to bring people together and to develop technology that can be used in human patients later on. So if you have ultrasound, you can basically stimulate very small parts of the brain. So you could go to a resolution of up to 1 cubic millimeter. Okay. So because you have a higher resolution, you also need a higher resolution connectome. You need a better idea where exactly to target. So not just what brain region to target, but what component of a brain region to target. And for this, we developed something we call super resolution diffusion tensor imaging, so structural connectivity, where we have 50,000 nodes. So this means that each brain region would have 500 to 1,000 nodes on average. So we basically take a structural image, we reconstruct the cortical surface. So we have 50,000 triangles on the cortical surface. So those are the nodes of the network. And then we use diffusion tensor imaging to look at connectivity between those nodes. And we then get a higher resolution of the system. So we can zoom in and zoom out of the adjacency matrix. So you can see when we zoom out the two hemispheres, left hemisphere, top left, and right hemisphere, bottom right. And now comes the tricky bit. Okay. So we can basically zoom in into the left hemisphere, and then we can zoom in further. Okay. So I have to keep my finger on here. And so what you basically see is that you have a modular organization within brain regions. Okay, so in the middle of the screen, you can see this area with two big modules inside that area. So this happens for many of the brain regions, that there is a modular organization within. And one thing we're currently testing is whether this modular organization is also linked to function. So if you think about the motor cortex, there was one part of the motor cortex for arm movement, another part for leg movement. So those might be separate modules that we can see here in structural connectivity. So we're currently testing that with combined fMRI data in motor tasks. One question is whether this high resolution connectivity is just an artifact. So we know that when fibers are hitting the cortex on the last mile, well, not last mile, last millimeter or so, they might change direction. Okay, so there might be some artifact in terms of the network that we are creating. In order to test that, we looked at connectivity between two regions. So I think if I use this, then people online can see it as well. So we have one area here, left superior frontal cortex and left superior parietal cortex. And along the diagonal, you can see modules within those structures. Okay. And then we can look at the connectivity between both regions. That's what you see zoomed in here. And if there would be some reconstruction artifact, we would expect that the connectivity between regions is more or less random. Okay. But this is not what we find. What we find is that one module in one region is connected to one module in the other region. So there's a clearly organized connectivity of this high resolution network. So this is all tested in healthy participants, but then we wondered whether this can give us some information in patients. So normally when you look at patients, you look at connectivity between regions. That's all people use in the field at the moment. But this might not be the most informative way to look at brain connectivity, because there might be local changes. There might be local cell death and local changes within a brain region before you see changes at the global stage. So we wondered whether this local information could be more informative than the normal global connectivity that we are studying in the field. For this, we looked at epilepsy patients, temporal lobe epilepsy patients, where the drugs don't work. So those are candidates that would undergo surgery. 33 patients and 36 controls. Again, we reconstruct the cortical surface and we reconstruct connectivity. And then we look at both kinds of connectivity, connectivity within regions. Okay, so only within regions, not between. And then the normal network where we have brain regions, 68 brain regions, and then fiber tracks between brain regions. And we found that the local information is actually more informative about the patients. So one example here is we try to predict a surgery outcome. So we have the good outcome. So that's ILAE score of 1 or 2, which basically means seizure-free or almost seizure-free. And 3 to 5 is that the patient still has seizures. And in order to do the prediction, we take connectivity before surgery. Okay, so we don't know anything about where surgeons are cutting. That's not part of our model. We just look at the baseline before surgery, where we measure structural connectivity. So we look at all those brain regions, and then we measure how many measures are actually different at the local level. So we can look at, for example, path length or clustering coefficient within a brain region. We compare that with what we see in healthy controls. And if it's significantly different, then this would be part of our overall measure. So we basically have the Cohen's d. So basically, this tells us how strong the changes are compared to healthy controls. And if we then look at cases with good outcome, you see that fewer brain regions show those local changes. On the other hand, if the effects of epileptic seizures are more widespread throughout the network, and you have a lot of changes throughout the network, this tends to be a bad outcome. So even if we remove those parts of the brain, for example, there are enough other parts of the brain that can then become starting points of epileptic seizures. So you would still have seizures after surgery. So we can then run a prediction model to predict what is happening. We have on the left the local network within brain regions, and on the right the standard global network, fiber tracts between regions. So you can see that the overall performance is a bit better with the local network. Okay, it's already quite good with the global network. But interestingly, we get a much better specificity, 90% compared to 75%. And this measures whether the local changes are better in correctly predicting bad outcomes. So you have a better prediction model in terms of predicting that surgery will not work for that patient. And of course, that would be interesting to know before surgery, right? I mean, if you have a high likelihood that the surgery doesn't work, you might not want to do it, or you might have to think about some alternative targets or some alternative way how to do it. Future questions are local network features better predictors for what regions should be targeted in surgery? So we might look at this previous picture and say, well, actually, this part of the brain should be removed because this shows really strong changes in that particular patient. So we might use this local information to predict what part of the brain to resect. But we could also use it as a starting point to decide where to stimulate with non-invasive brain stimulation or with implants. And it could also be a biomarker of disease progression. So what we see in this paper here is that the longer the disease progression, so the number of years with epilepsy, the more changes we see and the more widespread changes we see. So this could be useful early on as well. For example, for schizophrenia, you might be able to see changes 5 to 10 years before the onset of a psychotic episode, just by local changes in the network. So it's something to think about in the future, whether we can actually have predictions for later disease onset as well. This will become very interesting in the near future because we are getting an 11.7 Tesla human scanner for Nottingham. It's a national high-field MRI center. And because of that, we will have even better resolution below a millimeter. So we can get more information about local changes in the system. So we can get some better ideas what is actually happening during brain diseases. So this was all about targeting. But once we have a target, we want to understand how are connectomes linked to cognitive performance. So if we target a certain structure, can we also predict how cognition is improving? And I will make the point that it's not just important what information and how much information arrives, but also when it arrives. So if you look at information theory, you might think, okay, if you have a bigger fiber tract, if you have more streamlines, then more information is flowing, or you might count the information content in spikes and so on. But I will argue that it's very important to think about the delay. Okay, what you have in dementia, for example, is demyelination, and without myelin, you have a longer time it takes for information to propagate over fiber tracts, which means that information doesn't arrive at the right time. So this might be one component for cognitive impairment, to have changes and delays in the system. So let's start with looking at the spatial organization of the system, reducing neural wiring costs. So Klaus knows that part very well, but I'll just talk about it anyway. So there's this idea that the brain would minimize the total length of all connections in the system, because there are costs for establishing connections, so for example, having pioneer fibers and then establishing links between different parts of the brain. But there are also costs for signal propagation to get back to the resting membrane potential. So there are costs involved with having long connections in the system. And this has led to the idea of component placement optimization proposed by Christopher Cheniak in 1994. He was a philosopher based in the engineering department. So he basically found out about electronic circuit design, microchips at that point. And for microchips, you basically want to minimize the amount of wiring because you have higher heat dissipation, you have a higher loss of electricity if you have longer wires on your microchip. So he thought, well, that makes sense. The brain might do the same. So the idea is that whenever you rearrange two parts of the brain, you just swap the spatial position of them. The connections stay the same, but only the spatial position is swapping around. The idea is that always leads to higher total wiring length. So there's no way to swap brain regions around and get a better outcome. So this is an example here. We have those 4 nodes. You just rearrange A and D, you swap around the position of A and D, and you can see that the total length of the connections got longer. So the idea is that this always happens. However, looking at different data sets with better information about the spatial position of brain regions, we could show that that's not the case. So you can get a 30% lower total wiring length in rhesus monkeys. And if you go to C. elegans, you can even get a 50% lower total wiring length. So it is possible to get a better spatial organization of the system. So then the question is, why would the brain be organized in a non-optimal way? Why are we spending additional energy on connectivity? What you see here for the monkey is the optimal arrangement, so 30% reduced total wiring length. And this is the original arrangement. And if you look at the length distribution of fiber tracts in the system, you can see that those long distance connections have basically been removed by changing the position of brain regions. So those long distance connections are basically contributing towards this non-optimal arrangement of the system. And then the question is, well, what are long distance connections good for? What is the benefit of long distance connections? So of course, if you heard about small world networks, long distance connections can often be shortcuts in your network, so you can easily reach other parts of the brain. But there are other benefits as well. You can have synchrony of near and distant regions. So those two connected regions are receiving information at about the same time if you have a direct long distance connection to the target. So there might still be a slight difference, maybe 2-3 milliseconds difference in receiving the information, but it's more or less around the same time. There is reduced delay. So even in the best case scenario, you have an incoming action potential and the postsynaptic neuron is firing. This is introducing a 1 to 2 millisecond delay in terms of propagating the signal. So the more steps you have, the longer the total delay. You also have less cross-modal interference. If you want to go from the back of the brain to the front of the brain, you don't have to go through all the intermediate targets. So if you want to go from lateral interparietal area to the frontal eye field, basically transmitting visual information, you don't want to go for the auditory cortex. Okay. One thing that is omitted in textbooks is what you see in textbooks is that you have an action potential coming in, and then you have a release of neurotransmitter into the synaptic cleft. The sad truth is that in 50 to 90% of the cases, that's not what's happening. So you have an incoming action potential, but you don't have a release of neurotransmitter. So in order to have a neurotransmitter release, you need a sequence of action potentials to be sure that there is a release of neurotransmitter. So this also means that the more intermediate steps you have, the higher the likelihood that your signal will just be lost along the way. So there are many benefits of having this long-distance connectivity. And we know from Alzheimer's disease patients, for example, that they have reduced long-distance connectivity. And this reduction is directly correlated with cognitive impairment in patients, as measured by the midi-mental state examination. So this was the picture in monkeys. And then it took us a surprisingly long time to have a quick look at humans. All it takes is a good student dropping by, which was in this case Shui Chen, and help from Chris Hayward as well, looking at component placement optimization in humans. So we were using structural connectivity from 280 subjects of the Human Connectome project. And we did find improved spatial position is possible for all subjects. So you see for all 280, the histogram here, the change in total wiring length. So on average, it was possible to get a 21% reduction in the total wiring length. It's not 30% as in the monkey, but this might be explainable by the monkey only looking at one hemisphere. Whereas for the human brain, we had information about both hemispheres and connectivity between both hemispheres. So there were some more constraints in the human, and this is possibly the reason why we have a lower reduction that we can see in the system. And of course, if there would be no component placement optimization, all the values would be over here. Okay. So we have this clear pattern. So the old explanation is that long-distance connections are really useful. They reduce the number of intermediate nodes in processing paths, and they make it easier for information to go through the system. But in this paper, we were also testing a different idea, which is that the actual organization of the brain might be useful for network dynamics, for synchronization in the system. So we looked at synchrony and metastability. For this, each brain region is represented by a Kuramoto oscillator. So basically, each brain region can oscillate in a certain frequency. So we can determine whether we want to have 10 hertz or 20 hertz or 40 hertz. And then the phase can be different between those oscillators. We have connectivity between brain regions. So the strength of the weight or coupling strength is related to the number of streamlines. So the stronger the fiber tract, the stronger the coupling between oscillators. And we also have delays in the network. So the delays are based on the distance or the length of the fiber tract between the two regions. If you talk to mathematicians, they really don't like delays because it makes it complicated to run their models, which basically means that most models don't have delays in them. Which might be okay if you model activity at the local level. So if you look at activity within a cortical layer or within one area, delays are very small. So even if axons are non-myelinated, if you only have 1 millimeter to cross, which is the case for most neurons, you don't need to care about delays, right? But if you have information propagation between brain regions where you might have 10 to 20 centimeters in the worst case of fiber tract, then delays have a big impact. So we do have delays in our models. We then calculate the mean synchrony. Okay, so we look at synchronization over time, we calculate the average, and we look at metastability, which is the variation in the synchrony pattern. If we then look at the original network and the minimized network, we find that the minimized network, where we swap positions around and we try to reduce the total wiring length, for the minimized network, we have a higher synchrony. So you might wonder, well, isn't synchrony good? Wouldn't it be nice to have more synchrony? And no, not really. Okay, so you don't want the whole brain to be synchronous because that's what happens during epileptic seizures. It's not a nice state to have. So it actually makes sense for the brain to optimize for slightly lower overall synchrony in the system. What we do find is that we have lower metastability after optimization. To turn it around, we have a higher metastability for the original brain network. And this could have cognitive benefits. So if we look at the fluctuation, if you have a strong fluctuation of activity in your system, it makes it easier to switch to different brain states. Okay, you have a higher likelihood to switch to something else compared to having very low fluctuation in your signal. And this is something we see in dementia patients. So we had a paper in Brain where we looked at changes in EEG microstates. So you can measure activity pattern with EEG in patients and healthy controls. And then you can measure how long do patients stay in those states. And dementia patients stay in those states for longer. Okay. So it's a bit like they were stuck in a certain state and it takes them longer to get out of it. And this is exactly what we would expect. If you have lower metastability, it's less likely to get out of a state and move into a different state. So this tells you that delays might be important and spatial organization might be important. So as a next step, we wondered, can we actually use delay information or spatial information, and because of that dynamic information, as a predictor of intelligence? So we looked at the same data set, Human Connectome project, where we use topological measures. So that's what people normally use. You can look at hubs, for example, or you can look at path length and clustering coefficient. We had unweighted and weighted measures for those, and edge density as well. And then different subscores of intelligence, the full IQ, and then crystallized intelligence and subscores of fluid intelligence, processing speed, visual spatial ability, and memory. And you can see that some measures can, network measures can significantly predict intelligence, but at not many and not at high levels of significance. But if we then look at spatial features of the network, we get a much better prediction of intelligence scores. We have the convex hull, which is basically the volume underneath the wrapping paper. So if you wrap the whole brain, the surface of the brain just around the brain, and you look at the volume inside, so that's a good predictor. And we also look at the distances. So let's say you have nodes A, B, C, and A is basically either receiving or transmitting to nodes B and C. We wonder what is actually the distance between B and C. So if the distance between B and C is very narrow, we might integrate over the same module, for example, in the brain. And if the distance is very big between B and C, we might integrate between different parts of the brain. So we look at the global pattern of the distance between B and C, and then we look at cases where both of those connections are longer than the average, or both of those connections are shorter than the average connection length. And overall, we get a better prediction of those cognitive features. And one part of spatial organization is that it gives you an estimate of the delays over the fiber tract. So we then looked at dynamics, again, the same Kuramoto oscillator model with delays and connection strength. We can simulate in different frequencies, 10 Hertz, 20 Hertz, and 40 Hertz. And again, you can see a significant ability to predict cognitive subscores based on dynamics. So this is only a starting point. Maybe our dynamical model is not the best you could get. So maybe you need something that is a bit more detailed, maybe a Wilson-Cowan model with excitator inhibitor populations, maybe something even more detailed like what Changsong was mentioning to me, the spiking neurons and mean fields on spiking neurons and so on. But it is a starting point. So still the dynamic in many ways is as good or better than what we see in terms of topology. So we're currently testing whether we can use this approach also for patients. So we have data sets from Shanghai where we have schizophrenia data. So can we actually predict positive and negative symptoms in schizophrenia patients based on those topological, spatial, and dynamic features? So this will be interesting to know, because once we have a computational model that can give some information about cognitive impairment, we can use a computational model to predict how to stimulate the brain. So we can test hundreds of thousands of scenarios in a computer model to see which brain stimulation can actually give us the best improvement for cognition. based on those prediction models. So that's work in progress, but just something to keep in mind that delays might be very important in the system, not just topology. So let's look at focused ultrasound stimulation. It's a novel technology. The first international meeting about focused ultrasound neuromodulation took place in 2019 in Oxford. At that time, there were just 100 people in the room, so now there are a bit more people joining, so it's an emerging field. The first device for doing that in humans came onto the market in October 2019. We got one of those devices for Nottingham in 21. So you can see a higher spatial accuracy. So compared to magnetic or electric stimulation, we can also stimulate deep brain structures directly without stimulating the surrounding tissue. So we can be very refined in terms of what we are stimulating. The resolution could be very high, could be a cube millimeter. For the normal system, like for this system here, it's 1 cube centimeter. So it's not that high resolution, but we can directly go for deep brain structures and amygdala, thalamus, hippocampus, basal ganglia, or we can go for cerebellum and so on. It's non-invasive. Depending on the protocol, we can increase or decrease activity in the target. Okay, so we can excite or inhibit. Device can be used for humans or rodents, and it's possible to measure activity changes directly. So you can use this setup inside an MRI machine, so we can directly see changes in fMRI during stimulation, or we can use it with EEG. So it's really useful to see directly effects during the stimulation. How does ultrasound work? It's still kind of an open topic. So there are some papers out there, but there are many features that are not totally clear yet. For focused ultrasound, we have the transducer, which is a sound source. The sound source is curved, which basically means that the waves that are coming out of the sound source are converging at a certain point. And with this device, we can change where they are converging. So we can change some internal elements of the transducer to make it more curved or less curved. And by this, we can change where the focus is in the brain. So what happens is that you have some parts in the cell membrane, some channels that are mechanosensitive. Okay, so they're not reacting to voltage, but they react to pressure. If the pressure is high enough, they're opening. You have a second messenger cascade, calcium. So if you don't know what's happening in the brain, you just say calcium, and most of the time you're correct. And then you have an opening of other channels, including voltage-gated calcium and sodium channels. And then you can change membrane potential and make action potentials more likely and so on. So this is an older slide from 2020. That's a long time ago in the ultrasound field. So now there would be 2 or 3 times as many publications, but you can just see some examples how this was applied in humans already. It was applied alongside with TMS, with magnetic stimulation. You can see changes in 70 fMRI. It was applied to improve mood in healthy participants and also to improve scores for mild and moderate depression. There was a study for pain thresholds, traumatic brain injury, temporal lobe epilepsy. There was a study last year in schizophrenia patients from Shanghai, and that's a direction where we want to go into as well. I currently have 2 PhD students working on schizophrenia applications, so that's one way to go forward. So there are many different areas that were targeted and many different conditions that were targeted already. But one thing to keep in mind is that it's not enough to stimulate one target. You are really interested in what happens throughout the network, not just at the target, because the network response really gives information about what is the cognitive change that is happening in a patient. So this is data from Newcastle, where we have 1 monkey. Okay, so 1 monkey is not much, you might say, but that monkey went through a lot. So 25 ultrasound sessions and 27 control sessions. And we then wondered, well, what is actually happening throughout the network? For this, we tested for 3 different features. We have the strength of functional connectivity. So how is functional connectivity changing between the targeted region, which was left hippocampus, and other parts of the brain. So those are kind of global changes from the target to other parts of the brain. And then we can also look at local changes within brain regions. We have a fractional amplitude of low frequency fluctuation. So that's kind of spontaneous brain activity, and regional homogeneity. And this measures how similar the activity pattern in different voxels of a region is. Okay. So homogeneity would be very high if all the voxels of a brain region show the same time course. And if they're all different, then it's a lower regional homogeneity. So we can then look at what happens over time. For the global feature, we see a kind of narrow time window, and we also see this time window at a later stage. So initially, when you think about stimulation, you might think that you have an early peak, and then you have an exponential decay of the effect of stimulation. But this is not what you find. Okay. So you might find that at the target, but if we look at activity throughout the network, the pattern is more complicated. So we see this peak in terms of functional connectivity changes 15 minutes after stimulation. And if we look at the local measures, so basically looking at activity patterns within those regions here, we see a more diffuse time window. So some areas are starting earlier than others. So the idea would be that you have more or less rapid change at the global level, but then at the local level, some areas react earlier than others. And one thing we find is that this can partially be predicted from the distance between regions. So if regions are nearby, you have a relatively fast feedback loop. So it's faster to build up effects and then get above the threshold to what we see here. Whereas if regions are further away, it's a longer feedback loop, and it takes a longer time to build up changes and to be detectable. So if you have 3 monkeys, it's a population study. So we had 3 monkeys from colleagues in Oxford, the data here, and 3 different targets: so anteromedial, prefrontal cortex, superior motor area, and posterior ACC. And if we just look at one area here, we look at effects throughout the network, so other parts of the brain, and we see different time courses of the effects in terms of functional connectivity changes. So for one other area, you have an early peak. For another area, you have a peak somewhere in the middle. And then at the bottom, you have this area that is a bit active at the beginning. You have an early peak and then a bigger peak later on. So very complicated, right? But this has a big impact on how you analyze the data, because one lab might have a measurement 20 minutes after stimulation. So they get the first red line. They say, oh, you know, the first area is really, really active. Another lab measures after half an hour, and they say, well, the first area is not active at all. I don't see anything. And then another lab is looking at the third red line, maybe 50 minutes later, and they say, well, this area here, we don't see anything, no effect. Okay. So those results really indicate that it's not enough to take a snapshot of functional connectivity, but you really need to look at the time course of functional connectivity. You potentially need to observe over a longer time to really understand what is going on. So we then tested this in humans. So that was the first study in Nottingham with healthy participants. We have a baseline session measuring MRI and resting state functional connectivity. We have a stop signal task. So we have this green circle, and you're supposed to push a button. So you have the go pathway when you just push the button, and the stop pathway where you have this X sign appearing, which means you should inhibit the button pressing. You should not press the button. And then we have a session where we have ultrasound stimulation, and then we measure functional connectivity again, resting state functional connectivity, and then we do the test again. And we basically do the test 1.5 hours after stimulation. We use K-Plan. So K-Plan is a software package that allows us to estimate how much pressure we actually get to the target. We have 2 different targets, the white thalamus and the white inferior frontal cortex. And those are some parameters. The ones that might be interesting for you is that we stimulate for 80 seconds, and the duty cycle is 10%, which basically means we're effectively stimulating for 8 seconds. Lots of gaps in between. Okay. So you get 8 seconds of ultrasound stimulation, and you do see cognitive changes 1.5 hours later. But let's start with functional connectivity. So functional connectivity is measured. So you have ultrasound stimulation, then we get people out of the room. 50 meters away is the MRI. We put people into the MRI machine. We take anatomical scans, and then we have our resting state fMRI scan. And if we look at functional connectivity changes for stimulating the inferior frontal cortex, you see that different areas show different patterns in terms of changes in functional connectivity. What you see here is in blue, reduced functional connectivity. So you have a reduction in functional connectivity compared to the control for inferior frontal cortex stimulation. On the right-hand side, if you look at thalamus stimulation, for some regions, you have reduced functional connectivity in blue, but for others, you have increased functional connectivity in orange or red scale. So basically, the effect depends on the indirectly connected structures in the brain. So one reason for this might be that there are different cell populations in different parts of the brain, and depending on what populations you have, you might get a different effect of ultrasound stimulation. It's still relatively early days to understand this, but we do see differences in regions. So then 1.5 hours later, we look at the performance in the cognitive task. We find the same accuracy of performance. So people can mostly correctly stop when the red X sign is presented and so on. But what we do see is a faster reaction time when we stimulate the inferior frontal cortex. We don't see that if we look at thalamus stimulation. So we get an 8% faster reaction time. Maybe it doesn't sound like much, but given that the whole circuit is just processing visual information and then moving your finger, we would think it's more or less hardwired. So it is surprising to find 8% faster reaction time after stimulation. We can predict this change in reaction time based on biological changes, so based on changes in the functional connectivity between inferior frontal cortex and postcentral gyrus. And we can predict it based on physical features, which is the peak pressure at the target. So how much energy can we get to the target? If we can get more energy to the target, we see a stronger effect. So you have this mediation analysis here that both the physical effect and biological effect can predict the cognitive effect that we see in participants. Okay, I'm just checking. I might need to speed up a little bit. So in the future, what we really would like is to have something more portable. At the moment, the whole setup is 200,000 euros, and you can't take it home. That's in addition to having an MRI scan before everything starts. So we're thinking about some cheaper devices. So I'm on the advisory board of NewGear, and they developed a non-invasive ultrasound stimulator for the vagus nerve. So you just put it on like a headphone, and it's stimulating the auricular branch just behind the ear of the vagus nerve. So far, participants report being more relaxed and more calm. They also say they sleep better. So it might be something for anxiety disorders. It might be something for PTSD. It might be something for sleep disorders as well. So there are currently some tests on the way, and we have 6 of those devices in Nottingham. So we're looking forward to test this. At the moment, we are thinking about tinnitus. I'm co-supervising one PhD student, and she might look into tinnitus applications of this, but also looking at depression and schizophrenia. And another part is new navigation. So we have some ideas how to make new navigation more portable, so you don't need this 60,000 euro device just to let you know where the transducer is. So we're currently writing grant proposals on those ideas. But then we also need computational models to predict what is going on. So this is a paper that has been submitted about predicting the effect of optogenetic stimulation. So you basically have implants, you shine light onto parts of the tissue, and then you see changes in activity patterns. So this is basically demonstrating that a relatively abstract computational model, where we just have two populations, excitatory or inhibitory, can verify predictions that we see in terms of closed-loop optogenetic stimulation. So we can look at different waveforms of stimulation and look at the effect of the waveforms and of the gaps between stimulation and so on. And we see the same pattern in the computer model as we see in actual experiments in mice. We have a more detailed software, that's the Vertex MATLAB package, where we have individual neurons in our model. So we can have 10,000 neurons, they all have multiple compartments, they're all spiking, and you can look at changes in membrane potential and so on. So we can simulate different cortical layers, for example. So for this paper, we simulated the effect of electrical stimulation if we have an electrode implanted into the brain. And we can look at the effect of theta burst stimulation, for example. So in this example, we have this electrode reaching to this point here. It's coming diagonally, and what we see are changes in the synaptic weight of those neurons over here. So we see a change in particular for inhibitory Martinotti cells. And we can also use this model to make predictions about non-invasive electrical stimulation, where we have TDCS. And one problem with TDCS is it might work in one patient, but not in another patient. And we tested the effect of having a different direction of the electric field. So at the top, you see the direction of the field from the bottom to the top. So top is upper cortical layers, and bottom is lower cortical layers, like 5, 6. The opposite direction here, and then in this case, the field just goes laterally, left to right or right to left. And we can then, in the computer model, look at the effect of this angle direction of the field and the shift, DC shift in membrane potential, and the effect of using different intensities, so 2 milliampere, 4 milliampere, and so on, of electrical stimulation. And what you can see is that if you have this, this 180 degrees, okay, so that's going from top to bottom. If you want to have the same effect with a different angle, so with 135, you have to double your stimulation to 4 milliampere. Okay? So you need to put more energy into the system if you have the wrong angle. But of course, you can't do that without limit, because normally people just use 2 or 4 milliampere. So basically, there are some angles where you can't do anything. You can't get a proper response. And because the gyrification of the human brain is a bit like a fingerprint, so it depends a bit on the region. Some are more uniform, but some are more different between individuals. And this might be the reason why TDCS doesn't work all the time, because in one participant, you get this angle, and for another participant, the folding pattern is different, and you get a different angle. So in summary, ultrasound neuromodulation is preferred by patients and the general public. Effects are predominantly time-constrained and spatially distributed in brain regions functionally connected with the directly stimulated area. So just looking at structural connectivity is not enough. We can look at functional connectivity, and that's a better predictor of the amount of effects that we're observing. And those biological effects are indicative of cognitive effects. So we can look at functional connectivity changes and then predict cognitive changes like reduced reaction time. And the effect persists for at least an hour. We don't know how long it persists, might be 5 hours or so, but we didn't check. The cognitive improvement can also be predicted from, in addition, biological, predicted from the physical effects. So the amount of pressure we get at the target. And finally, there's a first step towards predicting dynamic effects, so predicting what is happening in the network over time. At the moment, relatively immediate, but the idea is on the long term to look at changes that happen over hours or weeks or months to get a better understanding of how we can change the connectome for a long time. I would like to thank my team, current postdocs and PhD students, lab alumni, collaborators, and funders. And if you're interested, you can follow me on X over there. So thanks for your attention.